like to move it, move it. I like to move it, move it. You like to move it. I like to move it, move it. I like to move it, move it. I like to move it, move it. You like to move it.
So, welcome to the first installation of our skeletal muscle lecture. And it makes, of course, your ability to move it. It's just a fun little thing. Okay, let me get to our slide. So hopefully you have found your study guides and you are ready to learn about muscle physiology and how your muscles help to generate movement, or micro movement, which helps things like balance, which these gentlemen are doing. It's just a few steps above me on the yoga skill set. All right, forward, march, here we go. Why can't I see things? Okay. Number one on your study guide, let's talk about some important functions of skeletal muscle. So the big four ones are, of course, one, movement, two, posture. So as you are moving around in the world, right, none of your muscles are completely relaxed until you die. And even then, you get through rigor mortis. We'll talk about that soon. But you have all these micro adjustments from your muscles, holding tension that allows for things like posture, as well as stabilization of your joints. That one of the things that helps keep your bones together is your muscle tone. And then, of course, thermoregulation, because as we've learned about, the production and use of ATP generates heat. Your muscles use a lot of ATP, and so they generate a lot of heat, so they help to keep your body at its lovely 98.6, or perhaps a little lower, or perhaps a little higher if one is, got a fever going on, trying to get rid of a virus. Part two of that. So in addition to skeletal muscle tissue, muscle organs also have connective tissue, and describe why that connective tissue is necessary for muscle function. So the image you're looking at here is a foot. Here's the calcaneus bone, and this is the tendon that connects the gastrocnemius and soleus to the calcaneus. Oh, come on, brain. Nope, it's not there. Anyway, your heel bone, and that's the calcaneal tendon, also known as the Achilles tendon. You can see here it has ruptured. So sometimes when tendons go under great stress, they can actually rip or kind of tear off of their connections to the bones. That can happen with rock climbers. I've seen that happen, I've heard stories, I haven't seen it personally, but where there's so much tension on those pulley systems in the fingers with the tendons that they actually rip off of the bones. So why do you need connective tissue? Well, you've got to have something that connects this muscle tissue that can contract to the bone, because otherwise you just have muscle tissue contracting, but you're not moving any bones. You have to have that connective tissue to reach over and insert into the bones. So when we look at the organization of that connective tissue, so here would be the entire muscle. Looking at a single muscle fiber, that's also a single muscle cell. That's surrounded by connective tissue, and then lots of collagen fibers there. And then groups of muscle cells are wrapped around in another layer of connective tissue, and then the entire muscle is wrapped around in connective tissue. And all that connective tissue kind of comes together and forms the tendon, which is full of collagen fibers. And so that together is going to connect your muscles to your bone, which is what allows movement. All right, next one, muscles have lots of nerves and blood vessels. Why are these critical to muscle function? All right. So each muscle has a rich blood supply. So it has at least one artery, but of course that artery is going to branch as soon as it gets into the muscle, and it's going to have a rich capillary beds. Why? Well, your muscles move through a lot of ATP, so they need a lot of oxygen, they need a lot of glucose, nutrients for cellular respiration to happen. They're also going to produce a lot of carbon dioxide that needs to be taken away. So you've got to have this rich capillary beds inside of your muscles. And in order to move, each individual cell has to be excited. So when you look at a muscle, it's full of thousands upon thousands of individual cells, and each one of those cells has a neuromuscular junction, meaning each one of those cells has an axon terminal that plugs in basically with a muscle fiber and excites that single muscle cell. You need one of those for every single one of those muscle cells or muscle fibers. So rich nerve supply. If you didn't have that, muscles wouldn't contract. And here's just an overview of kind of going from the large scale growth to the microscopic scale. Right, so here you're looking at the deltoid pectoralis major, but if you zoom in on that, you start to get this idea of the muscle being comprised of different groups of cells. Muscle fiber is a muscle cell, has multiple nuclei, but inside of that single cell, you have several myofibrils. What's a myofibril? Well, a myofibril is a group of actin and myosin collected together in these things called the sarcomeres. And the sarcomere, as you have no doubt learned already from doing your mastering A&P, is a contractile unit of the muscle. So it's doing the work of shortening the muscle so that you can move. And all of that sarcomere, that movement is dependent upon myosin and actin. So let's talk about all of those things. And of course, if you look at a muscle underneath a microscope, you start to see these dark and light bands. We call that striations. So what causes those striations? Well, it's how those myofibrils or those actin and myosin are stacked on top of each other. Some places they overlap, some places they don't. So let's look at that. So the bands, you've got an I band. Let's get the band back together. All right, you've got the I band, the A band, and this H zone and this Z disc. What's going on here? All right, so from one Z disc to the next, so right, like they kind of look like zigzaggies. So that's a Z disc. So this is a single sarcomere unit. In the middle of our sarcomere is the M line. That's nice. M line in the middle. And where it's just kind of the M line and there's no actin. So the actin is the blue and the myosin is the red here. That's called the H zone. And the A zone is, or the A band, sorry, is where from one end of the myosin to the other. That's kind of what defines the A band there. And then this I band would be where it's just actin. So you've got the Z disc in the middle of that and then the actin in the middle. So we're going to talk about how the A band and the I bands and the H zone change from a relaxed state to a contracted state. So this sarcomere is in the relaxed state. And you'll notice even in the relaxed state, you've got to have the myosin and the actin overlap. If they don't overlap, then the myosin can't grab onto the actin and pull it. If they're not overlapping, that's basically an overstretched muscle and it can't contract. Okay, anatomy of a sarcomere. I'll let you read that while I stay hydrated. All right, let's learn some stuff. So as we're reviewing, the middle of that sarcomere is called the M line, right down through here. And what's anchored to the M line is the myosin, also known as the thick filament. So the thing with the little feet or heads, depending on how you like to think about it, that's actually doing the pulling force. And then part of what stabilizes this this myosin is this titin, this yellow squirrely little protein here, and that's anchored to the Z disc on the other side. And titin is actually pretty interesting. They're still learning a lot about how titin works and its physiological functions. But one of the things it helps the muscle to do is to relax. So as, hold on. You ever have those moments where you're like, I just said something that's not quite right. Come on, brain. It's still caffeinating, which I don't do a lot of, but this morning. Okay. Too much information here. Okay. Myosin, thick filament, and it is what's doing the contracting, the pulling force. It's anchored in place. Part of what helps keep the sarcomere organized is this protein called titin. Titin's also important. You notice it kind of has this coiled look to it. This helps prevent the sarcomere from overstretching. So imagine you're stretching a muscle. If you stretch it too far, like I said, the actin and the myosin will no longer overlap. And once that happens, contraction can't happen. So the titin helps to resist overstretching of the sarcomere. Other characters we have in the sarcomere are the actin, which is what gets pulled, and that's also known as the thin filament. And then there are a couple more proteins on that actin. So let's look at those. Myosin heads, we've got millions and millions of those. Here's the thin filament, or the actin. So coupled with that actin are two other proteins called tropomyosin and troponin. And tropomyosin is the long piece that kind of looks like a long orange thread here. And you'll notice that it's in front of these little holes on the actin. And those are representing the active sites for the myosin heads to connect. And so in the relaxed state, those active sites are covered by tropomyosin. And troponins, these tiny little yellow proteins that you see interspersed every so often, their job is they actually have the ability to change the conformation of the tropomyosin, and they kind of roll them out of the way when it's time for the muscle to contract. Well, what activates troponin? Nice job. Calcium, right? So you need calcium is going to bind to the troponin here, and that binding of calcium to the troponin causes the troponin to kind of roll that myosin out of the way, and then the active sites of actin are activated, and myosin can act. It can pull the actin closer to the M line. All right, so relaxed versus a contracted state. So up above, we've got this relaxed state. And notice that right here's our A band from one myosin tip to the next. And here's our contracted state down here. And what you should notice is the A band doesn't change. So the length of the myosin isn't changing, but our I band is getting smaller because the actin is overlapping more here versus here in the middle towards the M line. And so that basically causes that H zone to totally disappear, and our I bands get reduced, but the A stays the same because none of the actin and the myosin aren't actually getting shorter, they're just overlapping each other more. All right, so now we've kind of got the basics and the mechanics around, let's excite our muscle. So next learning objective, explain what happens when an action potential reaches a neuromuscular junction. What neurotransmitter is released? You already know, it's acetylcholine. Nice job. What does this do to the membrane potential of the sarcolemma, and include the movements of sodium and potassium ions? So we're going to watch a video, but what does that do to the membrane potential? Is it going to depolarize it or hyperpolarize it? Yep. What's the answer? Yeah, it's going to depolarize it, right? Similar to the neuron. Muscles have a resting membrane potential that's set up similar to your neuron, in that you have a sodium-potassium pump and some leaky potassium and sodium channels, and that's going to maintain a negative voltage charge inside of the membrane of the muscle cell. And when the action potential comes down through the axon is going to release acetylcholine. Acetylcholine is going to bind to receptors in the muscle cell, and that's going to cause depolarization of the muscle cell. But let's watch it in action. Enough of Aaron yakking. Why is this picture here? Well, this is just also to remind us that when we excite a muscle cell, right, you have thousands and thousands of cells in each one of your muscles, and we're exciting a single muscle fiber with a single axon terminal. So we are going to watch AP flicks, events at the neuromuscular junction. So remember, on mastering A&P, you've got this portion called AP flicks. Let me show you where to find that again. And we're going to watch 3 of these. And what's handy to know about where to find those on mastering A&P is there are some great multiple choice questions, some of which you might see again, to test your knowledge. Oh, I've already got it ready to roll. But just in case you need a reminder, how do I have so many things? That's not the button I wanted to push. Well, while I'm figuring out my buttons.
Hot girls all over the world. Original King Juliet for the Gizman.
All right, just in case you need a reminder where to find this stuff, we're in Mastering A&P. I'm going to hit the study area. And then here's where your labs are, Physio X. We're going to AP, A&P flicks. We're going to go to A&P, Anatomy and Physiology. And here we are. Here are the 3 little AP flicks we're going to watch for muscles. First one's events at neuromuscular junction. And as I was saying, there are some great multiple choice questions. You answer them, you submit your answers for grading. That's not me grading, that's the computer grading and letting you know really quickly whether you're on track or not. All right. Without further ado, events at the neuromuscular junction.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across a synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
All right, so just to make sure you know where you're at, where are you at? Where are we? This is the membrane of the muscle. It's called the sarcolemma because everything needs a special name. So this is the membrane of your muscle, sarcolemma. So the axon terminals up here, it's released this acetylcholine. What you'll notice is that 2 acetylcholine molecules bind to this ligand-gated cation channel, right? So ligand gating meaning a chemical has to bind there to open it up. If it's a cation channel, that means it must let through cations, which are positively charged ions, because petting a cat is a positive experience. And that's where we're at. So we are about to see the change in the membrane potential of the muscle due to the neurotransmitter acetylcholine binding to these ligand-gated cation channels.
Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative.
Or in other words, depolarize. So it's a little different than the nervous system, right? Because depolarization in your neuron is totally dependent upon the sodium rushing into the cell. Muscles are a little different. Sodium is coming in, potassium is also leaving, but you have a lot more sodium coming in than you have potassium leaving, and so you have depolarization occurring.
Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
So that's what happens at the neuromuscular junction. That all causes depolarization, and then we're going to have an action potential that gets propagated along the sarcolemma or the membrane of the muscle. How does it get propagated? Same way it gets propagated in a neuron. You have voltage-gated sodium channels and voltage-gated potassium channels. So depolarization is going to happen with the, hold on, I think I actually have an image. Slow down, Egley. Okay, so wave of depolarizations moving this direction, right? So in a polarized states, negative on the inside, positive on the outside, this neuromuscular junction has released the acetylcholine, which has opened up these ligand gated channels. So sodium is pouring in, potassium is trickling out. That's going to cause less negative or more positive on the inside, more negative on the outside. And so that is going to open this voltage gated sodium channel, just like it did in the neuron. And that is going to self-propagate down the entire length of the muscle cell, right? Because my muscle cells, they go the entire length of the muscle. I'm, of course, pointing to my bicep, but right, so my bicep, the muscle part of that, it's about, I don't know, about that long. So each one of those muscle cells is actually the full length of my muscle organ. And so, but there's only one axon there that's talking to that single cell. And so once it gets excited, that action potential actually has to travel in both directions down that muscle cell. It gets propagated the same way an action potential gets propagated in a neuron with those sodium voltage gated channels, which causes depolarization, and then repolarization, same thing is caused by sodium, potassium voltage gated channels. Okay, so what role do T tubules play in muscle contraction? And what is their effect on the sarcoplasmic reticulum? Okay, so what are T tubules? Well, here we have our axon terminal neuromuscular junction. This little arrow is showing you that the action potential is going to move in both directions down the muscle cell. And then the sarcolemma, or the plasma membrane, kind of has these infoldings called these T tubules here. This is a T tubule, this white thing, and this white thing here. They're not actually white and bluish periwinkle in your body. It's just trying to differentiate some things so you can see what's what. But you can see how there's this little tube here. So there's actually this infolding. So then the action potential is going to move down this T tubule, and then as the action potential moves down that T tubule, then it's that voltage change is going to cause a change in this periwinkle networky stuff, and that's called your sarcoplasmic reticulum, which is a specialized endoplasmic reticulum in muscle cells. What's getting stored here? That's right, the calcium. So calcium is getting stored here. It's going to be released when the action potential moves down the T tubules. Let's see it happen.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T tubules, are regularly spaced infoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T tubules make contact with the calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So that's the process of excitation contraction coupling. And one thing I want you to notice about that sarcoplasmic reticulum is its surface area. So you'll notice that you have this net of sarcoplasmic reticulum around a bundle of actin and myosin. So each one of those little red dots would be an actin, and each one of those blue dots, which one of those red dots would be a myosin, and the blue dots would be actin. And so around a bundle of those, you have sarcoplasmic reticulum, and it makes this network, lots of surface area. Well, why do you need lots of surface area? Because calcium, you got to have calcium being released and in short enough distance that it can actually have an effect, right? Because calcium is going to bind to the troponin on the actin. So imagine this calcium that gets released here has to travel through diffusion to this actin fiber, where it's going to bind to the troponin on the actin fiber. So it's quite handy that that sarcoplasmic reticulum has a lot of surface area and is folded around bundles of myofibrils rather than just in one spot in the cell or even just on the outer membrane of the cell, because that would take too long for the calcium to diffuse from the outer membrane of the cell to the center of the cell. So it's distributed throughout. Likewise with these mitochondria, they are embedded and distributed throughout the cell. Why? Right? ATP. You've got to have ATP available to the myosin fibers so they can do their power strokes. So lots of surface area of the sarcoplasmic reticulum and lots of surface area of the mitochondria. And actually studies have shown that in muscles, the mitochondria aren't little bean-shaped things, but they make more like a network that kind of fills in the space between the actin and the myosin. So your friend and companion in physiology, surface area. Right, next step, cross-bridge cycle. So we're going to explain the sliding filament mechanism of contraction. What does that mean? Well, that means, right, sliding filament, meaning that my myosin is going to walk along the actin. They should have called it the walking filament mechanism because they're not really, they can't really slide. It's more that the myosin is walking and pulling the actin. So we've talked about the I, H, and A bands, right, and which ones shorten. So now we're going to get into the explanation of the role of calcium in excitation contraction coupling. So we just did that. Calcium gets released when the muscle cell gets excited, and now the calcium is going to carry that, hey, we got excited message to the actual actin and myosin to allow for contraction. So we're going to explain the events that occur during cross-bridge cycles and the role of ATP in muscle contraction, and then talk about how that applies to rigor mortis. So we're going to watch our last AP flicks, which is the cross-bridge cycle, which is basically these myosin heads binding to the active sites on the myosin, using that power stroke to move the actin closer to the M line. The cross bridge
The
cycle.
contraction of a skeletal muscle generates the force necessary to triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
That's the cross-bridge cycle of the myosin head attaching and releasing the actin based on whether it's bound to ATP or to ADP and inorganic phosphate. And as we've talked about, right, the A band stays the same width. The H zone here is going to disappear because as the actin move in, this kind of lighter band that makes up the H zone is gone. So here we have the H zone. Here it is gone because we've got the muscle in fully contraction mode. And the I band, right, this lighter band is going to get much smaller as the actin moves towards the M line because the myosin heads are pulling that actin towards the M line. Okay, so things to note about this, as you just saw in the video, which says it much better than I can, is that this myosin head is going to go through this cycle of being attached to the actin, released from the actin, and then rebinding to the actin. And what it's doing and how it's moving is entirely dependent upon whether it's attached to ADP and phosphate or ATP. So when it is bound to that actin, it's still bound to the ADP and phosphate. Removal of that ADP and phosphate allows for that power stroke. And then once that power stroke has happened, that myosin head is going to stay bound to that actin. Now, here's where rigor mortis comes in. After somebody dies, their body goes into this stiff state that can last, depends, 8 to 18 hours is what I've read. And why? Well, once you're dead, you can no longer make any ATP, and without ATP, these myosin heads stay bound to the actin, and so all of those muscles stay contracted instead of relaxed. So thus rigor mortis. Eventually, bacteria start to come in and start to eat things, and that eventually relaxes the muscles. But initially, it's in this rigor mortis or stiff state because there's no ATP to release the myosin heads from the actin. Great, we did that. So factors that influence the force and strength of a muscle contraction. So you've worked with this in the lab, working with recruitment and summation. So here's a little review in case you want to hear it one more time in a slightly different way. So recruitment has to do with your motor units. And here's a reminder that what's a motor unit? Well, it's one axon and all of the muscle cells it innervates. So one axon can branch a lot, and you can have thousands of muscle cells that are innervated by one axon. Or it can just branch a little bit. Maybe you'll have 5 or 6 muscle fibers or muscle cells that are innervated. Either way, that is a motor unit. So here's the image of a motor unit from your textbook. You saw this in the pre-lab video, right? Here's a motor unit. It's innervating 2 muscle fibers. This motor unit is innervating 3 muscle fibers or 3 muscle cells. This diagram, of course, is incomplete. If it was complete, then every single one of these muscle cells would have a neuromuscular junction with an axon terminal. Right? In reality, most axon terminals have way more than 2 or 3 neuromuscular junctions, but who wants to draw all of that? So what's recruitment? Well, recruitment is, you might start with just recruiting 1 or 2 motor units in a muscle, and then the more force or strength you need, you're gonna recruit more or activate more motor units, meaning more action potentials are gonna get sent down more axons and causing a greater proportion of those muscle cells to contract. So this is a nice image from your textbook because it's a graph. Everybody loves graphs. So stimuli to nerve. So we're starting here and we're moving in this direction, and here's our stimulus voltage. So this is what you were playing with in your lab, is as you increase the voltage, it's equivalent to increasing the number of motor units that are being activated. So here, with a low stimulus voltage, you're not quite to the threshold stimulus. You've got to get above the threshold to allow that action potential to propagate down the muscle cell. But once you've done that, then you can increase. So you'll notice here in 1 and 2, if you look at the proportion of motor units excited, you haven't excited any motor units because you didn't reach the threshold. So what are we looking at here? This is representative of a cross section of a muscle. And then as they're getting darker, that means that a motor unit within the muscle is getting excited. And what you'll see is you're increasing the stuff's in the way. All right, and here's tension, meaning the amount of force that's being generated by that muscle. So notice if we don't get above that threshold stimulus, none of the motor units are excited. We don't get any force, right? Because we're not getting an action potential that's moving down the sarcolemma to talk to the T tubules, which is going to release the calcium. But if we increase the stimulus voltage, meaning we increase the amount of voltage in the lab, in your muscles, it would be we're increasing the number of axons that are excited and sending an action potential down to the neuromuscular junction. As we increase that, we're going to increase the number of motor units that are excited, and we're going to increase the amount of tension or the amount of force that that muscle can create, right? We're talking about a whole muscle organ here. And at some point, we're going to reach maximal contraction. And why do we reach a maximum contraction? Well, because dun, 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 we have excited every single muscle fiber in that muscle. Now, in reality, in your body, that doesn't happen very often. The only time that happens is if there's a kid underneath a truck and you're the only one around that can lift that truck up off of that kid. In those instances, there is basically a break in your brain that prevents you from activating all of your motor units all at once in a muscle, because that can cause a lot of damage and strain, like on that connective tissue, that tendon that's holding that muscle in place. Those can get really damaged if you have too much pulling force on them, which can happen if you activate all of your motor units all at once. So even when you're at the gym and you're like really trying to lift as much weight as you can, there's a place in your brain that's like, hey, bozo, you can't do that because you're going to cause damage to your body. So we're going to prevent you from activating all of your muscle fibers at once. But in cases of extreme danger, that brake can come off of your brain and you can lift the truck up and pull the kid out. And then afterwards, you're probably going to have some damage to your muscle cells and your tendons, but you know, the kid's alive. So totally worth it. So maximal contraction here is happening when you're maximizing the number of motor units that can be stimulated within a muscle. So once again, recruitment, we're talking about whole muscle scale. And there's this fun thing with recruitment that has to do with the size principle of recruitment. So first off, we've got to recognize that not all skeletal muscle fibers or skeletal muscle cells are the same size. Some of them are larger than others. What makes one larger than another one? Well, the number of myofibrils in them, or the number of actin and myosin in them. So the more actin and myosin I have within a single muscle cell, the bigger it's going to be, right? Okay, that makes sense. So when we look at this graph, we're looking at time and tension, meaning the force that the muscle is creating. And what researchers have found is that initially when you start to use a muscle, you recruit your smallest muscle fibers first. And that has to do with, those are also the most easily excitable muscle fibers. So you start to use those motor units first, the ones with the smaller muscle fibers. And then as you need more force, as your body users are like, oh, we need some more work. This is actually heavier than we thought it was. Then you start recruiting medium fibers. And at the end, when you really need a lot of force at once, then you're going to recruit your larger muscle fibers. Okay, summation is another way that you can increase the amount of force that's being generated by a muscle, but now we're talking at a different scale. Summation is within a single muscle cell rather than the whole muscle organ, which is what recruitment's about. So now we're in a single muscle cell and we're looking at the frequency of the action potential, or the frequency, how much time is lapsing between action potentials from the neuromuscular junction. In other words, are we exciting the muscle once every minute, once every second, or like 500 times a second? I don't know if that's possible, but just throwing out some examples. So of course, as we increase the frequency with which this muscle cell is being excited, then the force of that muscle cell increases. So here's a single muscle twitch. We can't really measure that in vivo in your body, but that's something that can be measured in the lab, which is what you did with your lab simulation. So a single stimulus is going to cause a single twitch or a single contraction of a single muscle cell. And so here it's going through contraction and then this relaxation phase, right? So that's a single action potential coming from our neuromuscular junction. So what's summation? Well, summation is saying, well, we're gonna cause a action potential to go down the neuron, send that, and that's gonna release the acetylcholine, which is then gonna talk to those ligand-gated channels, which is then going to depolarize our sarcolemma, and then cause what? Oh yeah, release of calcium from that sarcoplasmic reticulum. And then that's going to start to cause contraction of our muscle. And then that calcium starts getting pulled actively. Well, that's kind of fun. Starts getting pulled actively back into that sarcoplasmic reticulum. And when there's less calcium around, well, what happens? When the calcium gets removed, then it releases its binding to the troponin. When it releases its binding to the tropomyosin covers the actin again, and it can't bind to the myosin heads. And so the muscle cell starts to relax. But notice it's not totally relaxed. That means there's still some calcium out there in the cytoplasm. So if before all that calcium gets packed back into the sarcoplasmic reticulum, we have another stimulus coming, that stimulus is going to cause more calcium to be released. So something that's important to recognize is when we're looking at this image, and the neuromuscular junctions release this acetylcholine, and the action potential is starting to move down our cell and go down the T-tubules, and that causes those calcium voltage gated ion channels to open up. With a single action potential, we're not releasing all of the calcium that's in the sarcoplasmic reticulum. There's still some left in there. So it's like we open the door for a little bit and let it maybe a few, 10, 20% of the calcium out of the sarcoplasmic reticulum, and then the door gets shut again. So not all of the calcium gets out with a single stimulation. However, if we increase the number of stimuli without letting the calcium being pumped in, we're letting more and more and more calcium out of the sarcoplasmic reticulum. And with more calcium being released, then more calcium is binding to the troponin, and more tropomyosin is being moved away from the active sites of the actin, and more myosin can bind to the actin and cause power strokes. Remember, the force or the tension of a muscle is all about the number of myosin heads that are doing this active power stroke. So the more actin active sites that are available, the more myosin can pull on those myosin filaments, and the greater the tension of the muscle. And that's all made possible by calcium. The more calcium you have, the more, the greater availability of the actin sites, and myosin can pull it. So that eventually you get to this maximum tension. And if you have enough, if you have enough stimuli all together, right, you get into this state of tetanus, which we don't see in the body, but we can see in the lab. But the state of tetanus would just mean that we've maximized the amount of calcium that's been released from the sarcoplasmic reticulum, and all of the myosin heads that are there that could possibly bind with actin and do power strokes are doing that. And that makes our maximum tension within a single cell. Okay, couple more slides for part one, and then we're gonna take a break. I'm gonna take a break, and then we'll do part two. Maybe you'll take a break too. Maybe you've already taken a break. I don't know. Hope you're doing well. This is fun. What is this? Another graph. Another graph. What's this one about? Length of sarcomere in micrometers, right? So we're looking at an increase in the length of the sarcomere, and here's the tension developed in percentages, meaning the amount, like if you put it on a scale, the maximum amount of tension or force that can be made based, dependent, right? Dependent upon the length of the sarcomere. So what we're looking at here is in this corner, you've got A, B, C, and D, and that is relative to this A, B, C, and D. What does this mean? Well, notice with D, the actin and the myosin are not overlapping. If they're not overlapping, then you can't generate any force from them. So this would be a sarcomere that's overstretched down here in D. B and C, the actin and the myosin are overlapped a bit, but not as much as at A. So we need some overlapping of the actin and myosin in order to maximize the amount of force that can be generated by our sarcomere. But if we already started a bit too contracted, then there's not as much space for the actin and myosin to go, right? There's less pulling power of our myosin on our actin if we were already starting somewhat overlapped. So in other words, there's kind of this sweet spot of just the Goldilocks principle of just enough overlap, not too little, not too much, but just enough that we can maximize the amount of force. And for the most part, that's kind of where our muscles are when they're relaxed. They are ready to maximize their amount of force. But this is kind of fun. This is one of Mish's favorite questions. And that's this. Explain why one of the goals in arm wrestling is to pull the opponent's arm towards yourself. So before you actually start the arm wrestle, right, with this thing, you actually want to pull your opponent's arm towards yourself a little bit. So what are you trying to do? Well, you are simultaneously causing D to happen in your opponent's arm. So you're stretching theirs out a little bit, so they're not going to have as much tension. And you are maximizing your tension. So you're getting your muscles in this C and B zone so that you can have much more tension than your opponent. Pretty clever. Try that next time you're not doing social distancing. I guess arm wrestling doesn't work with social distancing. Anyway, try that next time you're arm wrestling with another human. Okay. I'm going to hit pause, and we'll lecture 2 will come back to differentiating among isotonic, concentric, eccentric, and isometric contractions of the muscles, and talk about how muscles get their energy and what happens when we exercise muscles. Until then.